Welcome to this explainer. Today we're zooming way in, right into the invisible microscopic metropolis inside your cells. It's a bustling hub of constant activity in there, and we're going to spotlight one of the absolute unsung heroes of molecular biology.
So here's a question to kick things off. Right at this very moment, as you're listening to me, what is actually keeping your genetic code from just falling apart? Seriously, without one very specific enzyme working around the clock, our DNA strands would be dangerously fragmented. And, that would immediately lead to massive genetic errors and total cellular failure.
Well, meet that lifesaver, DNA ligase. Scientists universally call this the cell's molecular glue, and it is literally the ultimate biological fastener. Its whole job is ensuring the absolute integrity and continuity of our DNA. Whenever there are disconnected fragments of DNA, DNA ligase steps right in to join them together, making sure our genetic blueprint stays perfectly intact.
Okay, let's dive into Section 1, how DNA ligase works, the chemistry of the bond. Now, to actually seal these breaks in the DNA backbone, ligase forms a highly precise connection called a phosphodiester bond. It does this by linking the 3' hydroxyl group of one nucleotide straight to the 5' phosphate group of the one right next to it. But setting this glue takes energy. Now, what's really interesting here is where that energy comes from. It actually depends on the organism. In bacteria, DNA ligase usually grabs NAD+ as its power source. But in our own eukaryotic cells, and in many viruses, the enzyme uses ATP to seal those little molecular nicks. So, same exact job, just a different battery. Moving right along to Section 2, the cell's maintenance crew.
So, the enzyme's first massive job happens during DNA replication. When a cell divides, it obviously has to copy its DNA. But this process is, well, it's a bit messy. While the leading strand is copied in one continuous go, the other side, the lagging strand, is built in this really disjointed way. It's synthesized in short, discontinuous pieces called Okazaki fragments. Without DNA ligase, you'd basically just be left with a pile of unlinked fragments. The enzyme swoops in and perfectly stitches these Okazaki fragments together to create one continuous, flawless DNA strand.
But hey, replication is definitely not the only time we need this glue. Our DNA is facing a constant, relentless barrage of daily threats. We're talking UV radiation from the sun, chemical mutagens, environmental toxins, plus just your standard replication errors. It's practically a war zone in there. So, when damaged sections of DNA are removed and replaced, DNA ligase acts as the heroic repair crew, sealing the breaks to finalize the repair.
Ultimately, the whole essence of this enzyme is preservation. By repairing DNA damage and sealing strand breaks, DNA ligase helps preserve the overall stability and integrity of the genome. Oh, and it's also a key player in genetic recombination, where DNA segments are exchanged between molecules. That's absolutely crucial for genetic diversity and maintaining our chromosomes.
All right, Section 3, types of DNA ligases.
Just like you might have wood glue, superglue, and maybe craft glue in your house, complex eukaryotes like us have several highly specialized versions of this enzyme to divide up the labor. We've got DNA ligase I, which is the replication specialist, joining up those Okazaki fragments we just talked about. Then there's DNA ligase 3, which mainly handles standard DNA repair pathways. And finally, DNA ligase IV. This is the heavy-duty responder. It tackles severe double-strand breaks and is actually essential for the development of our immune system.
Let's zoom out a bit for Section 4, the biotech revolution, biotechnology's secret weapon.
Once human scientists figured out how to hijack this natural cellular maintenance crew, it literally triggered a revolution. Modern genetic engineering relies heavily on two primary tools working together. On one hand, you have restriction enzymes, which act as molecular scissors to cut DNA at very specific sequences. And brilliantly illustrating the other half of the equation, we have DNA ligase, the molecular glue that pastes those fragments back together. Because, you can't build anything new if you only know how to cut.
This exact cut and paste dynamic is the basis for recombinant DNA technology. It's a straightforward three-step process. First, you cut the DNA using your molecular scissors. Second, you produce compatible ends on the DNA. And third, you use DNA ligase to join those fragments, often from entirely different sources, into a brand new recombinant DNA molecule. This is exactly how we insert genes into plasmid vectors so they can replicate inside host cells, which is the foundation of gene cloning.
The real-world applications of this biotech are just massive.
Because we can harness DNA ligase, we can produce life-saving recombinant proteins. We can develop genetically modified organisms. It's the backbone of cutting-edge gene therapy research, and it's heavily utilized in modern vaccine development. The discovery and application of this enzyme quite literally changed the entire landscape of medical research and biotechnology.
Which brings us to our final topic, Section 5, when the glue fails, medical significance.
We have to ask, what happens when this essential natural glue stops working? When there are defects in DNA ligase activity, the genome essentially loses its stability. You get impaired DNA repair and skyrocketing mutation rates. And this isn't just some abstract biological concept, it leads to severe inherited disorders and a greatly increased susceptibility to cancer. If the breaks in the DNA aren't sealed properly, the cellular instructions get corrupted, and that's exactly how diseases like cancer can take hold.
Which leaves us with this final, really profound thought. If a defect in DNA ligase can lead to cancer, understanding exactly how this enzyme functions is precisely what helps scientists develop new, targeted therapies. By manipulating our own cellular glue, how might we unlock the future of targeted cancer treatment? It turns out, the key to our biological future might just rely on mastering the molecular glue that holds us all together. Thanks for joining me on this explainer, and keep questioning the incredible microscopic world around you.